The central dogma of biology is DNA being transcribed to create mRNA, and mRNA is translated to create a protein. DNA replication is constantly occurring throughout our body and is the unofficial first step in the central dogma of biology. Replication is a strand of DNA being copied and new strands of DNA are created. This occurs in the nucleus, with the help of an enzyme called helicase. Within the helicase, the old DNA strand is split and complementary bases are bonded to each strand using DNA polymerase. The bonds are adenine bonds to thymine and cytosine bonds to guanine. DNA polymerase is an enzyme that bonds the new bases to the old. DNA is an antiparallel double helix, meaning that one strand is called the 5' strand, as it has 5 carbons on the starting end, and the other is the 3' strand, as it has 3 carbons. The 5' and 3' strand are copied simultaneously, but because the 3' strand is a 3' strand, and replication happens 5' to 3', replication must occur by piecing the new strand together using Okazaki fragments, named for the scientists who discovered them. Replication creates two DNA helixes. Both helixes have one new, one old strand. This is called the semi-conservative replication. The next phase of central dogma is transcription.
Transcription has 3 stages. They are initiation, elongation, and termination. Initiation starts when the RNA polymerase, with the help of a basal transcription factor, binds to the starter region of the DNA strand. The starter region is a sequence of bases that will tell the RNA polymerase to start transcription, and it is found upstream from the coding section of the DNA. The double helix is then opened within the RNA. The next stage of transcription is elongation. This stage consists of RNA polymerase going along the DNA, reading and creating a complementary strand of mRNA to the template strand. However, the RNA strand will not have the base thymine. Instead, adenine binds to uracil. The mRNA strand is created 5' to 3', and transcription finally ends when the RNA polymerase comes across the termination sequence in the DNA strand. The RNA polymerase then unbinds from the DNA strand, and the mRNA strand is separated from everything. The final stage of the central dogma is translation.
Translation. mRNA to protein. Translation is when the mRNA strand is fed through a ribosome and read by tRNA to produce a protein. There are 3 types of RNA. mRNA, which stands for messenger RNA, which holds the sequence for the protein. tRNA, which stands for transport RNA, which brings the amino acid to the ribosome. And rRNA, ribosomal RNA, which binds to the mRNA to stop it from being read wrong. The mRNA has a sequence, 3 bases long, called codons. These codons code for a specific amino acid. There is one codon that tells the ribosome to start coding, which is AUG, and 3 codons to tell the ribosome to stop coding, which are UUA, UGA, or UAG. The start codon always codes for the amino acid methionine. tRNA has a complementary codon, the anticodon, which is one end of it. The other end has the corresponding amino acid attached. When tRNA enters the ribosome, it enters through the A site, bonds to the mRNA on the P site, creates a peptide bond with the former protein, breaks the bond with the mRNA, and leaves the ribosome through the E site. Translation is complete when the stop codon is reached and the mRNA strand is released from the ribosome and the protein is off to be folded and shipped to where it needs to go.